Hello. Do you think having introns or the whole splicing mechanism is actually of any advantage? It doesn't seem like, does it? It is unnecessary to have those because it occupies so much space. They take up the energy and material to build them and retain and have them in all of our genes. But why does evolution have come up with these, or how is it still, or are there any advantages of having introns? Well, at least we'll try to justify that there is an advantage, and that's what we will discuss now. One of the advantages is supposedly alternate splicing. I do tend to agree with that. And alternate splicing is something like having different forms of splicing in a given gene. Say, for example, we have what we know, of course, is if we know that there is a gene and it has, say, one intron, or say we say two introns because we are talking about alternate splicing. So there are two introns, 1 and 2. They should be removed. And normally we would get a simple exon, spliced exons and that's the mature mRNA, and it will give rise to one protein. And this is what is typical of majority of the genes or many of the genes. But can we get more than one type of gene when from one cistron, or more than one type of protein from one gene? It's a question here. And that is what will be the topic of alternative splicing. In essence, what we see is one gene can give rise to two different types of proteins. When we say two different types, there should be at least one change in the amino acid sequence. So even if this protein that I'm showing here is having a sequence like this, say of 6 amino acids, and in this position, in the third position, say there was tyrosine, and in the second protein, and in the third position alone, if there is serine, then the rest of the sequence is same. Still, these two are two different proteins. So that is what we will try to see as a part of alternative splicing. As is depicted here, alternate splicing we have here. One other thing that we need to remember is that about the open reading frame that forms upon splicing of the exons. So here we have a start codon, and we would have had stop codon. This is in DNA, and when mRNA is produced, we would have start codon and stop codon. These are the 5' or leader regions and 3' untranslated regions. So here is a start codon and here is a stop codon, and these exons should be spliced, and as a result we would get a mature mRNA which has start and stop codons and continuous codons there. And that is obviously what is meant by open reading frame. So now we can have in some cases one exon, the whole exon 2 might be skipped, and only exon 1 and exon 3 are spliced together, and that's where we have a different protein, a different mRNA, which upon being translated will produce a different protein. A protein that is produced from the normal splicing that we know, which constitutes exon 1, 2 and 3, would produce a protein, say something like this, right? Whereas if one exon is skipped, we would only get a protein with 2 domains, assume like that. So these two are different proteins. Similarly, we may also have an exon extended. In the cases, some of them, what may happen is there may be a pseudo splice site like sequence, which means this would have been the 5' splice site, and this would have been the 3' splice site. So splicing should occur between these two. That is 5' splice site and 3' splice site, or the end of exon 1 and beginning of exon 2. But because there might be, in some cases at least, there could be sequences that are similar to that of the 5' splice site. As a result, what may happen is splicing could occur between these two. So the fragment, the intron fragment might be left out. So it might be considered from here till here as one exon. That's why we call it this as exon extended. If splicing occurred between these two, then we would have a small fragment of intron left out or considered as part of exon. So we call that as exon extended type. There is another one which we call it as intron retained, because the whole intron that is present here, the intron that is given here is used up or left as such. No cutting transesterification reaction has occurred. So we have exon 1, 2 and 3 and this whole intron also as a part of the mature mRNA. And obviously when this is translated, we would get a different type of protein. And in the last one, we have, until now we did discuss about several types, and here we have a situation where this is the start codon, and I repeat, this is the stop codon. What if there is a stop codon here, right? There could be a stop codon there too. And in those situations, the outcomes upon translation would also be different. Say, for example, we will see, so if there was a stop codon here, and there is also a stop codon here, and assume that splicing has occurred, and we would get exon 1, 2, and 3. We could also have exon 1 and 2. We could also have alternative exons, exon 1 and 3. I'll just write the numbers for reference. This is exon 1 and 2, and here we have exon 1 and 3. At the end of splicing of the exons, what we should always be aware is that there should have been an open reading frame, because the ribosome doesn't care what it is translating. All it needs is an open reading frame. The start codon, it will read continuously the codons, and whenever it comes across the stop codon, termination of translation occurs. So here we have the normal type, and we have a stop codon here and also here. So if ribosome was translating, it would have given a protein with 2 domains, just for understanding. In the second scenario, if there is a translation occurs, it will also produce with a protein which consisting 1 and 2 domain. So in the first case, like in here, it doesn't matter if the exon 3 is present or not, because the ribosome would have translated, and it would have come across the stop codon, and there the translational termination would occur. And in the last scenario, we have exon 1 and 3, so we might have a different type of protein. So this one is called as alternative splicing. So it will have either 1 and 2 or 1 and 3. Okay. So did we talk about why not have an exon, a mature mRNA with just 2 and 3, exon 2 and exon 3? The problem is that there is no start codon. Does it have an open reading frame? It does not have an open reading frame as intended here, because there is no start codon to start at the appropriate place. If ribosome were to bind here and it did scan until it comes to a start codon, a protein could be produced probably, which is actually not of the function. So in alternative splicing, what we need to pay attention is to ensure that there is a open reading frame that has formed, and which is probably has a function to the cell. We'll see a few examples later. Thank you very much.